everybody. Thank you for being here. I'm Barbara Burtness. I'm the Associate Director for Translational Science, and it's really my honor to be kicking off our distinguished lecture series for this year at the Cancer Center by having the privilege of hosting Dr. Rafi Ahmed. So Rafi is a professor in the Department of Microbiology and Immunology at Emory. He's also the director of the Emory Vaccine Center, co-leader of the Cancer Immunology Research Program at Winship Cancer Institute, and an investigator in the Emory Center for AIDS Research. He is really a pillar of the immunology revolution that has impacted the care of cancer patients. He's, as I said, a professor in the Department of Microbiology and Immunology, a member of the National Academy, and a world-renowned immunologist. His work in the past decade has been highly influential in shaping our current understanding of memory T-cell differentiation and antiviral T and B-cell immunity. His goal is to use this information for vaccine development and insights into virally mediated cancers. A major area of focus for him has been identifying cellular molecules that regulate the generation and maintenance of CD8 and CD4 T-cell and humoral immunity. One such molecule is mTOR that his lab recently identified as a major regulator of memory CD8 T-cell differentiation. Before we get to hear him, and I promise you he's a brilliant speaker, I do want to have the opportunity to present a plaque to you. So I'm going to come up. So this honors your role as a distinguished lecturer here. Okay, thank you for coming.
Thank you. Did it fall off? Yeah, okay, yeah, okay. Okay. Yeah, what I'm missing my glass. Okay, can you all hear me? Okay. Thank you, Barbara, for this very kind introduction and for inviting me. Did we succeed on our third try or the fourth try? We've been trying to arrange my seminar, and something or the other happens. And I know, Barbara, that you have a flight to take, so please don't miss your flight because of me. Feel free to leave. All right, so I'm going to talk to you about T-cell exhaustion. And I thought I'd start by really asking the question, what is T-cell exhaustion? What does it really even mean? I think in the early days, there were very few labs that were studying T-cell exhaustion. And although we didn't know everything, I think we kind of knew what we were doing. But this has become now a very important issue in many areas, especially in cancer. And I think so. I thought I'd spend the first 5-10 minutes of the talk really kind of taking you through some historical observations that were made that really first pointed out the phenomena that during chronic viral infections, the T cells are not very functional. Just give you just a brief summary of that and then bring you up to date in terms of what we currently mean by T cell exhaustion. And then I will spend some time describing these very critical resource CD8 T cells, or the stem-like cells, which maintain CD8 responses during conditions of chronic infection. After that, I'll switch to describing our recent work with HPV-positive head and neck cancer. And if there is time, I'll present a summary of our recent work with the combination therapy of PD-1 plus IL-2. Okay. But if I don't, Barbara, I can just stop after the HPV one. Okay. Yeah. So the question is, what is T-cell exhaustion? So the history of this really started first, I would say, in the 1970s. when people who were studying mouse models of chronic viral infection, in particular from LCMV, but also from mouse hepatitis virus and a strange virus called lactate dehydrogenase virus, which is also used to be, now nobody uses it, but was a model for chronic infection. And there were these observations that based on functional assays, that the T cells were not as responsive as one would expect. And interestingly, at that time, the major T cell assay that was used was chromium release, chromium 51 release. And what was noticed was that when you took T cells or you took spleen cells from during the acute phase of infection, there was good killing of the target cells. These would be virally infected target cells. But when you took spleen cells from a chronically infected mouse, there was minimal chromium release, that is minimum killing. That really was the first beginning of those. And of course, later on, cytokine assays also confirmed that. And then these observations made in mice, chronic infections, were quickly extended to several human chronic viral infections. To HBV, people again found that people who are HBV carriers, again, it was difficult to detect good CD8 T cell responses in the blood. Similar with HCV, some of the herpes viruses, and in particular HIV, some of the early beautiful work done in human infection showing T cell dysfunction actually was in HIV. Okay. And then we were at this stage, the question still remained, is the decreased CD8 T cell response that we are seeing by some functional assays, is this due to deletion of the virus-specific cells, or are the T cells still present and truly dysfunctional? And this was an important question because often it was thought that when we don't detect a T cell response, that either the T cell response was never generated or the T cells had been deleted. And we should thank Mark Davis for the development of the MAC tetramer technology. This paper by John Altman, Michael McKaiser-Williams, and Davis in Science in 1996. This was a major breakthrough because this allowed us to, for the first time, to actually physically visualize the T cells that we were studying. Not just a functional assay, but now actually you could see it. And this allowed actually our lab and Rolf Zinkernagel's lab, and these were the papers published by Alan Zayak, who was a postdoc in the lab, and Gallimore was in Rolf Zinkernagel's lab, to show that when you compare an acute infection, LCMV infection, with a chronic LCMV infection, in both instances, you find tetramer positive cells, they are there. But when you do functional assays, like an ICS assay, intracytokine or ELISpot, then the functionality of those cells is much less. Okay? And this was the clear first demonstration that the cells are present, but that they lack or are not as functional as the memory cells or effector cells you get during acute infections. And of course, this was very quickly extended to human chronic viral infections and to cancer. So then the next question that was addressed by us and by many other people in the field was, what is the gene expression profile of these exhausted cells versus the acute cells? It was shown that actually they're strikingly different. The gene expression program of functional memory cells and exhausted cells is very different. This was work that John Weary did in 2007. Many other people followed up on it, extended it to humans. It was also shown that PD-1 is a major regulator of the CD8 T cell exhaustion. And so at this time, basically, we knew that the gene expression profile of the exhausted cells was very different from what you see in terms of memory cells. It was known that there are PD-1 and also many other inhibitory receptors that regulate T cell function. But what was not known until about 5 years or so, until 2016 or so, was really a clear definition of the different subsets which are there. So exhaustion wasn't a single cell that was functionally exhausted, but basically there was very interesting heterogeneity of the pool of exhausted T cells in terms of the functionality. So this was work that was done in my lab by Sage and Im, and also Utschneider published a paper around at the same time, working with Dietmar and Werner Held. And there was also a paper by Lin and Yi. These three papers came out in 2016, and they defined what I'll refer to as the stem-like CD8 T cells. These cells are also often referred to as TPEC cells, as standing for precursors of exhausted cells. And there are several interesting features of this cell. And actually, my talk today really is about this cell, okay? And I'll tell you more about this also in the context of HPV. So there were very unusual features of this cell. The first unusual feature was that even though this is in the conditions of a chronic infection, these cells are mostly quiescent. They're not rapidly dividing. There's some slow self-renewal. And also their location was interesting. Even when you have a situation where virus is in multiple tissues, both lymphoid and non-lymphoid, these cells tend to be mostly in lymphoid tissues. And within the lymphoid tissues, they are in the T cell zones. And that's an interesting point because these cells express CXCR5, but they still don't go to where the B cells are. They're remaining more where the T cell zones are. So they're in the T cell zone, and they're also resident. That is, they're not circulating. So this is the population that's within the spleens and lymph nodes of a chronically infected mouse and has been shown in humans. They're also in the tumor environment, but again, they are mostly not proliferating, and they're quiescent and also resident. So what do these cells express? They, of course, express PD-1, because PD-1, as has been shown by many people, is a marker that comes up upon TCR stimulation. If you take a naive T cell, you stimulate it in vivo or, or in vitro, within 24 hours, PD-1 comes up. It's a marker that comes up immediately upon TCR activation. So these cells are seeing antigen, they're PD-1 positive. But this stem-like population also expresses TCF1 and expresses BCL6, has high levels of co-stimulatory molecules. Other than PD-1 and a little bit of TIGIT, that doesn't have too many other inhibitory receptors, these cells. They have no effector molecules. They express interesting chemokines and chemokine receptors. They have CXCR5, so they respond to CXCL13. But they also have sufficient amounts of CCR7 that they can respond to CCL19 and 21. They express interesting chemokines that bring DCs around them. They're one of the few cells that express XCL1. XCL1 attracts CDC1, the CXCR3 positive DCs, so they are right next to. So basically, they have a nice niche that they've surrounded themselves with. And what these cells are doing is that in a slow rate, continuously, they're generating these transitory effector cells. So the step in this differentiation is downregulation of TCF1, upregulation of TIM3, TBET. They start proliferating, express effector molecules, and go out into circulation. And then eventually, these cells will get what we call, and others have called, the terminally differentiated cells with minimal proliferative capacity. So if you look at, to go back to my question, what is exhaustion? So basically, exhaustion is all three of these cells at any given time. The ratios of these three can differ. The stem-like cells usually are a very small percentage and only in the lymphoid tissue. They'll be about maybe 10 to 20% in the lymphoid tissue. If you go to non-lymphoid tissues, there's hardly any, less than 1 to 5%. Effector cells usually are also on the lower side, not a large number of them. Can be about 20%, 10%, especially in an established long-term chronic infection, they're only about 10%. Most of it are these more differentiated exhausted cells. So when we and others in the past were looking at all three of these, because if you use a tetramer, you sort all three of these populations, we were mostly measuring this cell in terms of the gimish that we had. But there are clearly cells there which are pluripotent and functional. And you have this effector cell that's being generated. And then you have this terminal differentiation. So the answer to the question is that T cell exhaustion is actually an ongoing immune response. It is highly regulated by inhibitory receptors and also by the immunosuppressive myeloid cells that surround that. So it's suppression by the myeloid cells, inhibitory receptors on the T cells, plus probably some other key inhibitory cytokines that are doing it. But it's active and it's ongoing because you're always generating a low number of effector cells from that stem-like population. And the key cell to maintain this whole exhaustion program, if you want to use that term, is the PD-1 positive, TCF1 positive, TOX positive. And I prefer to use the term resource cell because this is the cell that's feeding the T cell response under conditions of chronic infection. If you don't have the cell, the system just collapses. That is because the effector cells are short-lived, they go further, become exhausted. Those cells also have the lifespan is not very long. So what does PD-1 do? Basically, PD-1 changes the ratio of these cells. When you do PD-1 blockade, you get much faster differentiation of the stem-like cells to give you more of these effector cells. So the main thing the PD-1 does in terms of changing it changes the ratio. That is, you get many more effector cells, which will contribute to the killing of these target cells. And then eventually it will go into this thing. So when are these cells generated? Actually fairly early in the program, you get generation of these stem-like cells. And then, as I told you, this is what keeps the engine going. The next slide is just giving you a quick look at the gene expression program, again, published by many people multiple times, but just to give you a flavor of how these three cells differ in gene expression. Again, all three, so this is using a MHC class I tetramer to sort the antigen-specific cells and then doing single-cell RNA-seq. You see these three subsets. You've got the stem, the transitory, and the more exhausted or the terminally differentiated. All express PD-1, all express TOX. Only the stem-like cells express TCF1. These two don't. TIM3 is expressed by these cells and not that, not the stem-like. Again, 39, another inhibitory marker expressed by the more differentiated cells and not by the stem-like cells. I've spiked in here just what the gene expression program is of naive cells. So naive cells, of course, have no PD-1, they have no TOX, but they have TCF1. In fact, every good T cell has TCF1. Memory cells have TCF1. The stem-like cells have TCF1. And also you have TCF1 in all naive cells. TIM3 and 39 don't have it. I mean, not except the naive cells. If you look at effector molecules are not expressed by these cells. You have them in the differentiated, transitory, and exhausted. Again, granzyme B comes up. CD28 is higher in these cells. And CD127 is expressed by the stem-like cell. Again, CD127, as is well recognized, is essential for long-term survival of the T cell. IL-7 signals are essential. So if you don't have CD127, then a cell will not be persisting or surviving long-term. And it's the stem-like cells that have IL-7, as do as naive cells, of course. And XCL1 really only made by these cells, not by these or by the naive cells. I want to spend a few minutes asking the question, are these stem-like cells actually receiving TCR signals actively? And I've told you they express PD-1, so it suggests that they should be. But then we ask this question more directly by using NUT77 reporter cells. This is a transgenic mouse. where if the cells get TCR signaling, express NUR77, which is downstream of TCR signaling, GFP will come up. So this shows you the NUR77 staining, in this case, is really looking at GFP. So if you look at a mouse that has cleared the infection, and is a LCMV immune mouse, here's the tetramer staining. And of course, these cells have neither NUR77, minimal to none, and they don't have PD-1. But if you look at the antigen, same specificity, antigen-specific cells during chronic infection, you see that they're all NUR77 positive, and they're all PD-1 positive. If you then ask the three subsets, the three clusters, and we can subdivide it into the three clusters by using these markers, TIM3 and CD101. These are the stem-like cells, these are the transitory effectors, and these are the more terminally differentiated cells. And then you ask at NUR77 staining, you see that all three of them are NUR77 positive. So even though these cells, so all of these cells are getting TCR signals continuously. But in spite of the fact that you're getting the TCR signals, the stem-like cell is not dividing. There's only minimal proliferation. The transitory cells which have recently emerged from it are all mostly in cycle. So the recently emerging effector cells from the stem-like cells, at any given time, over 50% of them will be dividing because they're just emerging from it. The terminally differentiated cells, in terms of proliferation, are truly exhausted. They have minimal to no capacity to further proliferate. They don't do that after PD-1 blockade. They don't do that after any cytokines that you give. So these are not dividing. But this cell, which is very pluripotent and is able to divide after you give PD-1 blockade or other cytokines, under these conditions actually is quiescent. And then if you ask which cells express effector molecule, this stem cell expresses no granzyme B. Even though it's getting TCR signals, it expresses no granzyme B. The transitory cells, of course, have a lot of granzyme B, and the more exhausted cells also have granzyme B. So I find this quite fascinating, that this cell is getting TCR signals, we know that this cell is functional, pluripotent, and it can, after you do the PD-1 blockade, it will differentiate to give more transitory cells. But in its own state, it does not express granzyme B. Basically, think of it as a TFH cell, because the TFH, it has BCL6, it has in some ways hijacked some aspects of the TFH program. So when you have CD4 cells, they differentiate, you get TH1, you get TFH. The TFH cell, which has BCL6 and so on, it shuts down the effector program. So TFH cells will never have effector molecules. It's the TH1 cells that have it. So the stem-like cell has taken some interesting aspect of that biology. It certainly is not a TFH cell. It doesn't even go near B cells. It's not involved in helping B cells, but it has captured a very interesting aspect of TFH biology in its differentiation program. But when it then differentiates, you remove the PD-1 break. When it differentiates, now you get the TH1 type cells, which is your transitory effector cells, which express all the molecules that a cytotoxic T cell expresses. I think this is really quite interesting biology for us to work out. So the stem cell niche, so this is not expressing these things is actually an active program. It's not a passive program. It's not due to lack of TCR stimulation, not due to lack of antigen. That is the program. That is the transcriptional program that changes after you remove the PD-1 break or can do it by some other cytokines that you provide. Okay, so let me now get to human stem cells. Many people have shown this. Elegant studies done looking at neoantigen-specific cells in melanoma, in many other cancer systems have shown that you have these stem-like cells also in human cancer. We have shown this in lung cancer and head and neck cancer. I'll just show you couple of slides from lung cancer, just to tell you that, yes, you find these cells. And this was the paper we published last year, but I had also shown this in another paper earlier. But the more interesting thing from this study, in addition to showing the stem-like cells in lung cancers, was the location of these cells within the lung cancer. So if you look in the lung cancer studies, in lung cancer samples, these cells are not where the tumor is. They are away from the tumor. So on the tumor, you will find the more effector-like cells, the transitory cells, and you find cells which are the more exhausted or terminally differentiated. These stem-like cells are away from where the actual tumor is. And we found that some of them are actually in tertiary lymphoid structures within the tumor. And they're closer to the CD4 T cells there than to the B cells. So essentially, the similar kind of compartmentalization that we saw in the chronic viral infection model, that is, they're not where the actual virally infected cells are, but a little bit away from it. So like if you look in the spleen of these chronically infected mice, the major infection is in the red pulp of the F480 macrophages and other cells. These cells are in the T cell areas where there isn't much infected cells. So they're hiding this beautiful niche that they have. This keeps them away from the major, it's just part of their whole program that keeps them there. It's when they differentiate that they will send the effectors out there. And then there was a beautiful paper, much nicer paper than ours, paper by Neil Cohen, who did this, published this paper this year. And in this one, he shows that, again, in lung cancer, if you look at the stem-like cells, they are in what he calls stem immunity hubs, which are, again, removed from where the tumor is, and they're close to where the T cells are. In fact, he makes the analogy that these cells are found in areas that look like lymph node areas. Okay? And this is all within the lung tumor. He's not looking at a lymph node, he's looking right at the tumor. And we had done the same thing. We found that where you see the cancer, the tumor antigen, very few of these cells are there. They're always away to the side. Okay, let me now go to head and neck cancer. And one of the reasons we did this, Barbara, not because we knew anything about head and neck cancer, but because we wanted to look at antigen-specific cells. Because in our lung studies, when you're looking at total cells, and we did not have the bandwidth to be doing the sequencing and identifying the precise mutation in each individual and doing it. So we said, let's go to a viral-mediated cancer, where would be easier for us to identify the epitopes which are being recognized, and then we can use tetramers and look at HPV-specific cells in the tumor. So we started out by collaborating with Nabil Saba and Mihir Patel, and what they gave us was the following material. They gave us, in this study, we have now extended it and we have more data, but I'll share with you some of our published work. So basically, we had 17 patients who were HPV-positive head and neck cancer patients. These were all treatment naive. By what I mean is that they had not had previous treatment. They had just been diagnosed as having head and neck cancer. And we got from them at the time of surgery, we got the sample of the blood, and we got the primary tumor, which you all know is in the tonsils. And then we also got a metastatic lymph node, not a draining lymph node, but a metastatic lymph node. Yeah. Now, at this stage, we had no idea what responses these individuals were making in the tumor. And the tumor material, as you know, is very limited. So we first had to identify what potential epitopes were being seen by these 17 patients. So what we did to address that issue, we did the following experiment. So we took the blood from these HNSCC patients and then expanded these cells for 2 weeks, basically expanding the cells in vitro. And we used about 250 predicted HPV peptides. We, of course, included E6, E7, which is the canonical oncogene, but we also included E2 and E5 because there were some reports saying that one can get CD8 responses to E2 and E5. These were not in tumor samples, but in people who were HPV positive. And so we included E2 and E5. And then expanded this population, looked at reactivity, if we could expand any cells from these individuals, from the blood. We did first interferon gamma ELISpot, then did ICS, and then if the response was good enough, we then went ahead and identified the precise tetramer. So here is an example of one of the good responders. It's a very nice after the expansion. We're looking now at the pool of peptides, all four of them, all 250. We see a nice response. By ICS, you see very nice interferon gamma and TNF alpha production. And then by tetramer, again, very nice staining. This is the same tetramer, double tetramer staining. You see that. And interestingly, all 17 patients that we looked at were positive. So there is all of these individuals. There was no one that was negative. Some were higher, some were lower in the response, but all of them gave us a response. That is, there was some low number of cells present in the blood that we could expand. And then we ended up identifying several tetramers that we made. Note that E2 and E5 tetramers, there are several responses came from E2 and E5, some from E6. This is not by any means comprehensive. We had these tetramers. So we used these tetramers. Now we could go back. We knew which individual was responding to which cell based on the expansion from the blood. So we can now go back and do the stain of the TILs themselves. And the results were really, I never expected this. I did not expect to see that in these individuals, this is now no expansion. This is just staining the TILs with the tetramer. So here is PD1 on this axis, tetramer on this axis. This is a tetramer recognizing peptides 151 to 158 from the E2 is AO1 restricted. And the primary tumor, and here's the metastatic tumor. From the metastatic, the lymph node that also has the tumor. 3%, 0.1%. Here's another one, 5%, 10%. It's really quite amazing that these treatment-naive individuals who are coming in for surgery have such a vigorous response ongoing in their tumor. This is a, what if we use the same tetramer and look in the blood? Very low. The response is barely detectable. Okay. Because again, I didn't talk about this earlier, but in the we did some parabiosis experiments in our chronic LCMV infection mice to see which cells are in the blood and which will actually migrate. It's only the transitory effectors that will come out. So the more differentiated cells are resident at the sites of infection, and the stem-like cell is resident in the lymphoid areas. So the only cell that circulates are the recently generated effector-like cells. So it's not surprising that in the PBMC, there are very few. It's not zero, because you know that's where we expanded these cells from. So we know that the cells were there, but when you do a stain, the frequency is very low. I highlight this part because we often look at new antigen-specific cells. We want to look at tumor-specific cells in the blood of somebody who's not had any treatment. It's not that they're not there, but you'll have to look very hard. Okay? There'll be a very, very low frequency. So here is some example showing you the frequency of about six or seven different tetramers in different individuals. And you can see that very high frequencies ranging from 0.1% up sometimes even 10%. But in the blood, by this approach, below detection, i.e. below 0.02%. So a lot of cells in the tumor, okay, but very few cells in the circulation, antigen specific. Okay, so the next question, of course, was what do these cells look like? Do we have that stem-like population? So we did single-cell RNA-seq, and the answer is yes. You again find within the tetramer sorted cells. Now, this is all on getting the cells, isolating them directly from the tumor, and then doing single-cell-seq. And again, you have the canonical cell here, which has TCF1. It's amazing that XCL1 always goes with them. It wasn't just a mouse phenomena. Every human stem-like cell defined by us or by the others have shown that XCL1 is there, because again, this attracts CDC1. It's part of the niche that they create. And again, no granzyme B in these cells. And you have LEF1, which is another transcription factor you see in these stem cells, more IL-7 receptor over here, and again, very little perforin and so on. So we looked at about 15 or so tetramers in different patients, and all of them have the three subsets. So the blue is the more exhausted subset. This was in most cases the highest number, but all of them had this transitory population, which is shown here in yellow, and also had the stem-like population. So these three, so in these head and neck cancer patients, you have an ongoing response there. You have the stem-like cell is generating some effector cells, and these are eventually getting exhausted. It's a very active program ongoing. In this study, we also looked at the TCR sequences, and the TCR sequences are shared. At least the major clonotypes are shared between these three, again, establishing the lineage relationship between them. And the other interesting finding was, in some cases, these responses are very oligoclonal. In some cases, about 50% of the tetramer cells were a single clonotype. In others, a little bit more heterogeneity, but very oligoclonal. And I think you only see this oligoclonality when you have long-term chronic antigen stimulation, which has selected for certain TCRs. Okay. Then we asked the question, are these stem-like cells, the ones shown here in green, are these cells actually functional? Can we somehow demonstrate functionality in these cells? So for this, we sorted we didn't use tetramer here because the frequency is too low. We had to get as many cells as we could. We wanted to do the CTV labeling experiment. Frequency is not low, but the total numbers are not enough. So we enriched for this. So basically, we did a sorting strategy that would enrich for where the stem-like cells would be and where the more differentiated ones would be. So for this, we basically gated on PD-1 positive cells, and then used cells which were TIM3 negative and CD39 negative. So this would enrich for the stem-like population. And then the TIM3 positive, 39 positive would enrich for the more differentiated population. Then we cultured these, we labeled the cells with CTV, so we could look at division, and then stimulated them now we know exactly which peptide to use. So now we could stimulate with their cognate peptide, and you can see the results here. So we're looking here at 5 days, after 5 days in culture. This is the unstimulated, that is no peptide stimulation. Here is on this axis is the tetramer. Now we can stain with the tetramer. So even though we didn't sort with the tetramer, we now stained with the tetramers. We're truly looking at the antigen-specific cells. And what you see here is that there's minimal proliferation without the peptide. And here's the tetramer population here, no division. And if you look now here, after adding the peptide, with the more differentiated cells, very minimal to no proliferation. So again, the proliferative capacity of these more exhausted cells is very minimal. But look what happens here. These cells proliferate beautifully. So they have this ability, under the right conditions, you provide the peptide and they will proliferate. You get very nice proliferation here. This is a summary of all the experiments that we did. Minimal proliferation of the more differentiated cells, but very nice proliferation and CTV dilution of the stem-like cells. And what happens now? Do they differentiate into effector-like cells? Yes, they do. So if you now look at the stimulations of how the markers change, TIM3 comes up, granzyme B comes up beautifully. Okay. See, before there was no granzyme B in these cells. Granzyme B comes up. CD39 comes up. IL-7 receptor was there, goes down. Okay. So and you can see that these cells had IL-7 receptor, these cells didn't. Okay. As I've told you earlier, these cells don't survive that long. But after you generate the effectors, IL-7 receptor comes down. And of course, over here, not a whole lot is happening, except to point out that these cells were already TIM3 positive. These cells had granzyme B, whereas these cells did not. These cells had, had 39, these cells did not. All of these cells are RO. There's some confusion in the literature that this stem-like cells are RA. No, it's all RO. And this is not the stem-like cells described after acute infection. Everything is RO because they've all antigen experience. In fact, they're seeing antigen. CD25 comes up beautifully after this proliferation. And CD28, which I was pointing out earlier, it's only the stem-like cells that have CD28. And they require this co-stimulatory signal for the expansion that you see after PD-1 blockade. So it's the stem-like cells that have CD28, and interestingly, they're retaining it, at least in this shot, after the shot in vitro stimulation. Whereas the more terminally differential actually never had much CD28 to begin with. Okay, so this is what I was telling you, where they are located. So this is now stain of a tumor, of the HPV tumor. And you can see that this is where the tumor is based on the cytokeratin staining. Lot of CD8 T cells in this tumor. And if you look at PD-1, pretty much all of them, or most of them expressing PD-1. This is the CD8, there's the PD-1. And now you look at TCF1. They're here. This is where there was no tumor. Again, they segregate. They're not where the tumor is. Their progeny will go to the tumor, but these cells will be in these niches. So let me conclude this and kind of end on a couple of next steps that we're doing. So basically, we've identified 3 transcriptionally distinct states of HPV-specific CD8 T cells. And I didn't show you this data, but this is in our published paper. And they share the TCR. The same TCR clonotype is found in all 3 subsets. Again, consistent with showing that one is generating to give rise to the other. And I've shown you functional evidence also because you have this, this thing happening based on the TCR clonotype being shared. But this is the more important point. The cellular machinery, essentially, i.e. the PD-1 positive, stem-like or resource cells that are required for response to PD-1 pathway blockade, they exist in these HPV positive HNSCC patients pre-treatment, suggesting that PD-1 adjuvant therapy could be an effective treatment. The other interesting finding here was, although the study is limited, by limited I mean we don't have hundreds of patients, we found that E2 and E5 were the dominant targets, in particular E2. E2 is a major target. And this finding of ours has been subsequent, in the last, since our paper came out, 2 more papers have come out showing the same thing. E2 is a dominant antigen in HPV. And so far, there have been some lot of experiments done using E6 and E7 vaccines that have gone into many clinical trials, both for cervical cancer and maybe some for, but most of this has been focused on cervical cancer, maybe some also head and neck. But we would strongly recommend that if you want to do a therapeutic vaccination of either head and neck cancer patients, especially for head and neck cancer patients where we have this data, that E2 and E5 should also be part of the vaccine, in particular E2, which is a very, very dominant antigen. So I want to kind of end with a couple of, so this was our paper published by today, but I didn't say anything about our second paper that was published with it, which looked at B cell responses to HPV in the tumor. And they are also very strong. So within the tumor, we find HPV-specific memory B cells. We have antibody secreting cells. That is, there are plasma cells right in the tumor. which are secreting antibody to E6, E7, and E2, and E5. So there's a lot of active B cell response ongoing. So these are, to use the term hot, these are very, very immunologically active tumors. Both B cell and T cell responses ongoing to multiple HPV antigens, with, at least for the CD8s, real dominance of E2 in this response. So what we have started now, and I was talking to Barbara about this, a very small study, and this is ongoing. Took us forever to get it going, but we have a small study that's being done by Mihir Patel and Nabil Saba. It basically is an adjuvant study where we are giving head and neck cancer patients 2 doses of anti-PD-1 that we got from Roche, from Genentech and Roche. So they will get, before they have their surgery, they will get 2 infusions of anti-PD-L1. And then they will go in for the surgery. We've been collecting blood from these patients. The plan is to have 20 patients who will get the PD-L1 treatment prior to their surgery and radiation treatment and so on. Okay. And so far, 7 patients have been enrolled. And basically, blood is being collected, pre-treatment, pre-infusion of the PD-1, we'll get 1 blood sample, and then after that, and then at the time of surgery, we'll get the tissue sample as well as the blood. So 7 have been recruited into the thing already. 2 of them have actually completed their regimen. And the early data, it's only N of 2, but both these patients have shown a pathological response within this month of anti-PD-L1 treatment. We haven't done the T cell analysis yet because we have the samples there. We first need to identify, we need to do what we did with the previous time and see what they're responding to. But here I have a prediction. Now we will see more cells in the blood, is my prediction. Because they have gotten the anti-PD-L1 treatment, blood, these effector cells should come out into the blood. Now we will see cells in the blood. That's my prediction. So that's where we are, and I'll keep you posted. And this is I will stop after the next one. I'm going to skip dial 2, okay? And then, so far it's been all head and neck, and but I've been in discussions with several people about maybe trying to do something similar in cervical cancer. The question is, in cervical cancer, is it going to be all E6, E7 only? Will you also have E2 and E5 responses in cervical cancer or not? So we are just starting a collaboration. Actually, I'm also going to India next week, okay, for this. So we are lucky that the Emory Vaccine Center has a lab at a very nice institute there called ICGB. So we have and we've had this lab for many years. Our focus actually has been on dengue virus infection. But now we will start a program on HPV, cervical cancer, and this will be in collaboration with one of the largest and the best hospitals in India, the AIIMS Hospital in New Delhi. Okay. So hopefully in the next few years, we will have some data on what responses look like in HPV positive cervical cancer patients. Okay. I will skip the IL-2 part. I should skip it, right? I have time? Okay, I'll go through it quickly. Most of it is published, but I think it's relevant because there's some IL-2. Okay. All right, so I will go through this quickly. So this, I'm switching now to rational design of immunotherapy. So as I mentioned to you, there are 3 of these subsets there. So what can we do to increase the number of stem-like cells? Can we do something to change the quality of the effector cells that we get? And probably the hardest thing, can we do something to get those truly terminally different to do something? But what I want to talk to you today with IL-2 is improving the quality of effector cells. IL-2 doesn't do much in terms of expanding stem cells, and it doesn't do anything on the more differentiated cells. Okay, so IL-2 doesn't do anything with this cell. But what we found is that, that when you combine IL-2 with PD-1, and this was published a couple of years back, that we see a dramatic change in the transcriptional program of the effector cells. Basically, what we see is that when you do, actually, let me go to this. Yeah, let me go to this slide. Okay. So basically, this is what the previous data shows you, that when you do, if an untreated situation, you have the stem-like cells, you have the transitory effector cells, and you have the terminally differentiated cells. After you do PD-1 blockade, you get many more of these cells, okay, which eventually will go here. But when we did PD-1 blockade plus IL-2, we totally change the program of these cells. So these stem-like cells have tremendous pluripotency. That is, they're not fate-locked into the kind of effectors they will give. So this creates another opportunity, not only to just get more of the blue cells, but also to change their transcriptional trajectory and epigenetic trajectory to get much better effector cells. And that's what PD-1 plus IL-2 combination therapy does. Not only do you get many more cells, but they're transcriptionally and epigenetically very distinct. And what do they look like now? Now they start looking more like effector cells that you generate during an acute infection. So I think that this offers really very interesting opportunities in terms of further manipulating that stem-like population. So then this was the part that was, at least to me, more interesting, but I think more problematic for the field, okay, is what we found in the qualifier is that this is a mouse model. Okay? What we found is that CD25 engagement was important. As you all know, the trimeric receptor is there's the alpha chain, there's the beta chain, and there's the gamma chain. The signaling is through the beta and the gamma chain. The alpha chain, which is CD25, forms the trimeric receptor. The affinity of the trimeric receptor is log orders more than the affinity of beta and gamma. And so our results were that if we use, so we did 3 kind of experiments. Okay. One was if we used a cytokine that was mutated, that is, did not have binding to the alpha chain, we actually saw no synergy at all. In fact, that basically it was the sink effect. It just went and bound every T cell that was there, okay, and was not targeted properly. And then we also showed that if you do CD25 blockade, then the synergy was totally gone. That is, using wild type IL-2 and then doing CD25 blockade, completely gone. Okay. That the critical way that cytokine was working was binding to the trimeric receptor. Okay. Skip, yeah. Again, this is what I told you. But there are ways out of it. That is, you can still use, so as I told you, and also that I skipped data, 25 comes up very quickly on the antigen-specific cells in vivo after this combination therapy. 25 blockade abrogates this effect. But if you target it, so we also did a study using a molecule from Roche, which was an anti-PD-1 antibody, on which there was IL-2. This was the mutated IL-2. And this works also quite well. Okay. So now you're targeting it to the cells of interest, and then you see an effect. Okay. And that was published there. So where do we stand now with IL-2 therapy? So I think if you use, and unfortunately, the failure of many cytokines, IL-2 cytokines is consistent with our findings, is that if you use a free cytokine, that is just the cytokine, but that's mutated in the alpha chain to binding to C25, it's unlikely to work. And several phase 3 clinical trials have failed. When a free cytokine is being used and the CD25, either Either by pegylation, you block CD1 binding, which was the nectar IL-2, and other IL-2s where this was mutated. They have either stopped after phase 1 or have unfortunately gone further and stopped later. But targeted therapy is one option, and many people are using this now. Some are using CD8 targeting, but probably the PD-1 targeting is better. And currently, there is a clinical trial that Regeneron is doing, where they are also using a PD-1 targeted, but they're using wild type. So Regeneron, they just started a clinical trial in lung cancer and also other cancers. They're using what we did, but with the wild type IL-2. And actually, that might even be better. Yeah. The issue with wild type IL-2 is the toxicity issue. But I think if it's more targeted, it can probably be it can be better. But in addition, there's also now regulated IL-2s being used. That is, they will only become active after they engage with PD-1. That will bring in another safety aspect to it. So I think that this field is going to see a lot of interesting data. IL-2 is, as I put in the first bullet point here, it is the key cytokine for effective differentiation. There is no other cytokine that we have that is better in terms of generating good effectors. In our mouse model, we have tried many, many different combination therapies. And the one that's the most superior is the IL-2 plus PD-1. So I think that the field now, I think, is in a place where some attention is being paid to targeting the IL-2 and not just using free IL-2 which doesn't bind CD25. That basically is a washout because every cell has the beta and the gamma chain. So it binds to every cell. It's just a major sink effect. I think that strategy is pretty much gone. But targeting is a real possibility. And then additional safety can come by regulating it so that it only becomes active after it binds PD-1. So I think it's an exciting time for these studies. So to end my talk on the stem-like chronic cells, I showed you data on the chronic infection and in cancer, both human and many people, others have shown this, but also in autoimmunity. So now there are several papers that have come out that in autoimmunity, also you have this subset. Part of the Th17 pathway would be something bad. But again, you need the cell. The key message here is that without the cell, you cannot maintain a T cell response, whether it's for good or it's for bad. This cell is absolutely essential to keep the engine going. So a lot of wonderful people in the lab. I mentioned their names when I was discussing. Great collaboration on head and neck cancer with Nabil Saba and Mihir Patel, and many wonderful external collaborators. Thank you.
There are questions. Yes.
So thank you for the talk. You mentioned that it's effector cells that connect the stem-like cells to the tumor differentiated cells in the target tissue, but then you showed that there are stem-like cells in the tumor itself. Wouldn't that suggest that it's the stem-like cells that are moving and then differentiating?
That's a good question. We don't have direct evidence for that. Okay? It's a very good question. Okay? Whether they move before they differentiate, my prediction would be that they start differentiating before they move because that program keeps them there. The chemokine receptors changed. So I think it's the differentiation step that then brings them out. And the key differentiator actually is downregulation of TCF1. And then TBET comes up, all these other things come up. So the initial signals for differentiation are happening in those hubs. Okay? But then once they differentiate, part of the program is you go out, which actually is not that different from what happens to a naive cell after infection or vaccination. Okay? The initial signals are in the lymph node and the T cell zones. Once they become effector-like cells, they will come out.
Of the talk, I was wondering about the tumor-like lymph node niches that we see. Do you also look at other cytotoxic effector cells like natural killer cells? Are they different? Do you find stem cells?
Yeah, we've been very focused on, we've even been eroding CD4 cells. So we've been very focused on, because again, the tumor sample is small, the number of things we can do. So we've been very, very focused. But Andreas Wieland, who was involved in both of these studies, he's on the faculty at Ohio State now, and he's putting a focus on CD4 T cells and B cells.
Can I ask you, this work was all done in treatment naive?
Yes, all done into,
patients
yes,
with cancer anatomy.
yes.
If, you know, in head and neck cancer, a big problem is that we've removed all the lymph nodes in many of our patients.
Yes.
And yet I think from what you were showing us in lung cancer and from the fact that we see responses in people with recurrent disease, I assume that the effector T cells that we generate with pembrolizumab in a recurrent
Yeah.
patient also come. from
Yeah,
the
no,
T
that
cells.
you're raising a
So-
very, well, you're raising a very important question, is that when you do surgery and you remove all the lymph nodes, how many of these stem-like cells are remaining? I don't know the answer for that. You probably are removing a lot of them.
But
So I
then-
think it's very, you can't leave the lymph nodes there because for obvious reasons, but you are, I think, because where are these cells as far as we know? They're in the tumor within these niches that, you know, Neer had defined and other people and we have shown. And they're in the draining nodes. That's been shown by many people. So when you remove both compartments, you certainly are reducing the number of those cells. The lung cancer that I told you, that was not, there was no surgery, that was people getting PD-1 blockade.
I'm just saying that there must be, I'm wondering if there's a process that recruits these TCF1 positive cells into distant
Yeah,
metastases.
right. So that brings up interesting questions about how do you, in that setting, how do you increase the frequency? I mean, one thing would be therapeutic vaccination. And hopefully there's still some naive cells with that TCRs, which can be recruited. And I don't think it would be that all of the cells would be gone from the lymph nodes that you remove. Okay? So I think you may then need to provide some vaccination strategy for increasing the numbers. Yeah. Numbers, I think, do go down after you do it. I can't imagine them not going down, right? Because you're removing the reservoirs where these cells were.
Exhaustion is this mix of states. And in all
There probably are more states. Okay, I'm sure that somebody will find subsets of effectors and subsets of stem, but these are the three canonical subsets. Yes. It's also tox positive. Yeah. It's happening continuously because when these cells are differentiating, they're giving rise to the effector cells, which remain, I think, good for several, we don't know exactly, might be days or weeks they'll be there. And then eventually they will go to that terminal differentiated state. There's some people who think that they might be, depending on the environment, they might be generating more of the exhausted cells immediately. But our data is more consistent with it going through a early functional stage before they become exhausted. But both are possible. So in terms of when is each thing happening, once the system has kind of set in, basically you have all three of these cells. Okay. Are you asking the question, when are stem cells generated? Oh, if they don't respond to therapy, then many things could be wrong. The tumor could be more aggressive. But from a T cell side, I think when a patient doesn't respond, I think the number of T cells to begin with are very low. See, PD-1 does not create any new T cell. None of the checkpoint blockade strategies create a new T cell. They are trying to provide more differentiation from an existing cell.
Take one last question maybe from Brenda Amu, and then would it be okay if people come up with?
Oh yeah, I'll
yeah.
stay, yeah.
the HPV infection. Small percentage respond to malignancy. Is there something in the pre-malignant stage that there's a difference that some patients lose sometimes?
Yeah, that's a very good question. Again, in terms of how their initial responses were, how many cells were made. So, and then also the question, when do you generate these stem? We have a full story on that, but they generated very early, very early. So actually so I would say that in the HPV infection, that this cell which has some of these characteristics would have been generated very early during the infection. If the antigen clears, then it will not have this phenotype. Okay? It'll then start looking more like a memory cell. Okay? But this cell population is generated very early. And that's a paper that's coming out. I didn't want to include that talk in here, but it's really quite nice because the immune system is actually preparing for generating that cell before even knowing the outcome. I mean, the paper that we have in press, and that would be a different seminar, is that even in the acute infection setting, you generate the cell population in small numbers. Because at that time, you don't know, is the infection going to be chronic or acute? So there's this very early preparation by generating this. So this fate decision to give you this cell, which is, again, I emphasize granzyme B negative and having the TFH flavor, happens very quickly within the first week. And this cell is there even in the acute infection. Then you look later, it's gone. It's become part of the pool of the central memory cells. We don't know how much of that contributes to it, but that cell is gone. But if the chronic infection ensues, then that is the key cell maintaining it. So I think in the HPV, what you're asking, I think this cell was generated when they first got infected.
Thank you so much.
That's it.